Fermentation is a process of converting sugar acid or into acid or alcohol. In simpler terms, it involves using yeast to transform sugar into alcohol or using bacteria to produce lactic acid in certain foods. When biotechnology is incorporated into this process, a bioreactor is used for large-scale production. A bioreactor is a device that allows living cells or enzymes to utilize low-value substrates to produce valuable products. It is widely used in various applications such as waste treatment, fermentation, and the production of pharmaceuticals. There are two main types of fermentation processes: aerobic and anaerobic fermentation. In aerobic fermentation, the key feature is that it requires an adequate aeration. Uh, for example, a supply of sterile air, which is typically around 60 times the volume of the medium, along with an effective steering and mixing mechanism. While in anaerobic fermentation, aeration is not usually required, although it may be necessary initially for in-column buildup. The mixing device is often unnecessary because the gas produced during fermentation provides sufficient agitation. At the heart of the system is the fermentation vessel itself, which holds the culture medium, a nutrient-rich mixture that supports the growth of the living cells being cultivated. Everything entering this vessel must be sterile to prevent contamination, as even a small impurity can ruin the entire batch. One of the most noticeable components of a bioreactor is the agitator system. This consists a central shaft with rotating blades or impellers powered by a motor. The impellers play a critical role in ensuring thorough mixing so that every cell receives an equal supply of nutrients and in aerobic systems, oxygen. For aerobic processes, air is introduced through a sparger, a ring located at the bottom of the vessel that releases sterile air bubbles into the medium. The impellers then break these large bubbles into smaller ones, maximizing the surface area for oxygen transfer into the liquid. For the next topic about enzyme technology and its applications, Mr. Mazo will discuss it.